Okay, I'd like to begin and welcome everyone to the Botstiber Institute for Wildlife Fertility Control webinar. Thank you for joining us. Today's webinar presentation will be the use of fertility control to manage urban prairie dog populations. And our presenter today is Dan Salkeld of the Colorado State University. My name is Monique Principe, and I am the Managing Director of the Botstiber Institute. And also with us from the Institute on screen today, or up above, is Stephanie Boyles-Griffin. She's the Science and Policy Director for the Institute, and Rachel Soroka, who's our Program Assistant, who's up there behind the scenes managing backstage operation of the webinar. So after Dan's presentation, there will be a Q&A session. If you would, please type your questions in the Q&A panel, which appears at the right-hand side of your screen. You may have to open that screen and get access to that. And you can do that by accessing the menu at the bottom of your monitor screen. There are 3 dots, and it'll show you where your Q&A is, and you'll want to click that and open it. So all of our questions today, you please type them in there in that Q&A box, and Stephanie will read them out verbally, and then Dan will answer them. Please do not use the chat function, as it won't be monitored today. And if we don't have enough time during the actual webinar to get to your question, all of the questions are being noted. So someone will answer your question after the webinar. I think that's it for housekeeping. So I'm going to hand it over to Stephanie, who's going to introduce Dan.
Okay, good afternoon, everyone. As Monique said, my name is Stephanie Boyles-Griffin, and I'm the Science and Policy Director for the Botstiber Institute for Wildlife Fertility Control. And it's my great pleasure to introduce everyone to Dr. Dan Salkeld. Dr. Salkeld serves as a research scientist too for the Department of Biology at Colorado State University in Fort Collins, Colorado, where he teaches courses on ecology of infectious disease and One Health. Prior to CSU, Dan was a lecturer and research scientist in the program of human biology, Woods Institute for Environment and Department of Anthropology at Stanford University. He also served as a researcher at the California State University, Fullerton, and University of California, Berkeley, as well as for IUCN, the International Union for Conservation of Nature in Washington, D.C. He holds a PhD in ecology from James Cook University in Townsville, Australia, and a BS in zoology from the University of Edinburgh in Scotland. Dan has published numerous articles in professional publications and presented at conferences and workshops around the world. Currently, his research focuses on the intersection of disease ecology, conservation, and public health, and emerging tick-borne diseases in places like California. Finally, Dan is a BIWFC grant recipient, and the work he will be presenting today was supported by the Botstiber Institute for Wildlife Fertility Control. And with that, I will hand it over to Dan.
Thank you, Stephanie. Am I on? Is that, yes. That's right. Okay. And then... Okay. Is the screen being shared?
That looks great, Dan.
Okay. Thank you very much, Stephanie and Monique, for the introduction. So I will launch into this, and I'd like to start off just acknowledging my collaborators. So this was not done solely by me, obviously, but with collaborators at CSU, Jackson and Runt, and then also collaborators at USDA APHIS. So this has been a team effort, and those include Doug Eckery, Gary Whitmer, and Aaron Shields. And as mentioned, this was all made possible by funding from the Botstiber Institute for Wildlife Fertility Control, and so I'm thrilled and grateful to have that support and to be able to present this work. And finally, the places that we worked: Bluff Lake, Fort Collins Natural Areas, also the EPA and Colorado Parks and Wildlife for permits, and a great big crowd of fantastic volunteers for handling prairie dogs in the field. So this was all done with their help. So to give you guys an introduction, this work is on Blacktail Prairie Dogs. They're one of 5 species of prairie dog in North America. This is a map with a distribution of them here. They're very emblematic of Great Plains habitat, and they're also a keystone species. So what they do is they burrow, they make these huge complexes, as we'll talk about in a second, and they modify the habitat by foraging quite closely to the ground, and that alters the grasslands for other animals so that you get a large association between prairie dog presence and a great many others, and we'll talk about those too. One of the most important and charismatic is the black-footed ferret. So this is a black-footed ferret preying upon a black-tailed prairie dog. They're actually a little, the prairie dogs are a little heavier than the black-footed ferret itself. But these are near obligate predators of prairie dogs. And so without prairie dogs, you cannot have persistence of black-tailed ferrets. And they are one of the most endangered mammals in North America. So they were actually declared extinct very briefly until a remnant population was found in northern Wyoming by a man's sheepdog. And now there's a reintroduction program for black-footed ferrets run out of Fort Collins and some other locations in the western US. So the point being that without prairie dogs, we have no sustainable way to reintroduce black-footed ferrets. That is the most emblematic species related to prairie dogs, but there are many others. So in Colorado, burrowing owls, conservation status is threatened, and management efforts are directed towards retention of active prairie dog towns because of that impact on vegetation. And the burrowing owls actually use prairie dog burrows for their nests. And this, again, is another really charismatic species of the American West. And so we have to be looking at the underlying conditions for its survival. So that is prairie dog colonies or towns. So there's a whole lot of buy-in to protecting and preserving prairie dog habitat. And one of the instigating forces for this was an event in near Denver in Stapleton, where land developers were actually eradicating prairie dogs from some land to be developed. And they did this with poisoning, and we'll talk about that also. And locals were quite upset and perturbed by this situation, in part because the control methods could have health impacts for local people, but in part because they really enjoyed prairie dogs being part of their landscape and their dog walks, because it kind of encapsulates that western feel of the plains and outside. But like all things, there are also the bad sides for prairie dogs. So they are in their range highly associated with plague outbreaks. So plague being bubonic plague, the same disease that caused the Black Death in Europe in medieval ages. And that is now epizootic in the western United States. And plague, if in prairie dog colonies, will rip through the colony, causing a lot of devastation to the prairie dog population, and also occasionally acting as a potential source of human infection. So this is a Denver Post article from a recent outbreak in Commerce City, so quite an urban situation, and led to a lot of concert closures and people not being able to use the outdoor lands. And this is another downside. So prairie dogs, especially in urban areas, will can overgraze land if they're not allowed to grow in the colony. And so this is a dust storm from a prairie dog colony in Boulder, Colorado. And you can see the overgrazing has just led to this erosion of dust by the wind. And prairie dog density does tend to be a lot higher in urban colonies compared to natural and restricted populations. So the density of prairie dogs can actually be 5 times higher where they are kind of kept into small urban areas, and they can't actually colonize other areas or disperse naturally. And this has impacts for landowners. For example, in this conservation, this is Bluff Lake, where the numbers are high and they're causing erosion of this land. And this can have an impact also on housing if they are encroaching upon people's backyards and digging and causing damage to their structures. So there's a nice correlation between people who really support prairie dog conservation and those who don't. And the people who don't are often those who are living right on the borders of prairie dog habitat. The current solutions for prairie dog management are kind of imperfect and a little lacking. So there's non-lethal control. One is to capture prairie dogs and remove them and relocate them in other habitat. And that has its pros in that you are allowing them to continue and exist in nearby habitat, but often it can fail in terms of success. And it is majorly limited by the availability of land for it to relocate. So recently you have to get special permissions to even move prairie dogs across county lines because neighboring counties are not happy about prairie dogs being relocated. Barriers have also been used, so you can put up plastic sheeting so that it prevents the prairie dogs from being able to see and move outside of the current zones. But those are not always efficient, and often you can see prairie dog burrows arriving underneath the barriers. Lethal control is also used, so poisoning is one way to do that, or fumigation, and that can be effective, but it also has concerns in that people do not like that being used, especially in urban areas where there is close contact with human populations. And it also leads to the problem of it's not greatly sustainable. So poisoning or fumigation may reduce the population, but often the prairie dogs will then reproduce compensatorily so that their reproduction rate increases to take advantage of the empty spaces. And so you get a very fast recovery of the prairie dog populations. Raptor perches is also another way. So you actually encourage raptors to use the areas around prairie dogs and hopefully they increase predation. But that also tends to, it's not worked brilliantly and is a very slow burn approach. So the work that I've been involved in was asking whether we can use an immunocontraceptive so that we can control their reproductive output and hopefully have a win-win situation that we can maintain sustainable, controlled prairie dog populations and yet reduce so that they have the benefits of being intact wildlife in the West and acting as a keystone species, but remove the cons of it all so that you reduce overpopulation and overgrazing and overdispersal into nearby lands. So the immunocontraceptive that we adopted and tested was gonocon, which is a gonadotrophin releasing hormone. And essentially what it does is interfere with the reproductive hormone cascade. So it stops animals from actually entering the breeding cycle and reproducing. It's being successfully used previously on other species like white-tailed deer, California ground squirrels, Norway rats, etc. And lab studies have shown it that it doesn't seem to have a huge impact upon the health of the prairie dogs. And so it looks like it might be a good way to manage prairie dog populations sustainably. So what we did was essentially to test this in a real world setting. So we captured individual prairie dogs in the fall of 2018 on 3 different prairie dog colonies. And we injected gonocon 0.4 milliliters into the upper thigh of adult females. And then we had some reference sites. So we treated them with a sham vaccine, so saline without the gonocon, so that we could work out whether the procedure was having the impact or not. We used pit tags, so those are transponder tags that are put into the scruff of the prairie dog, just like people use for cats or dogs for their identification, so that we could then recognize the individuals afterwards. This is Jackson doing some work, and I know some of you will be eagle-eyed we were just catching, this was a cottontail rabbit, it's not a prairie dog, but we were just taking advantage of the camera and the situation to show how we capture them. So these are traps, tomahawk live traps. We use a handling comb so that they can be restrained, and we can treat them without the use of anesthetic, which allows faster recovery and release. So we adopted a matched pair protocol. So we have 2 matched pairs in Fort Collins. So you can see here up on the just near that Horsetooth Reservoir, there are 2 matched pairs. So these are done, but they're nearby with a reasonably similar prairie dog density. We have one in southern Fort Collins, and then we have a site in Denver. It's one site, but it used the far ends of the prairie dog colony. So you can see here about 10 to 30 is the region of or the sample size of prairie dogs that we captured each. And so we looked at the success of gonocon application in a couple of different ways. First, we used visual count ratios. So we looked for and counted the numbers of adults and juveniles as they emerged from burrows in May, June of each year. So at that point, you can tell the juveniles, they're much, much smaller than the adults. And so we just did counts with scans and binoculars. And we corrected the observations by putting out painted mason jars so that we could work out our efficiency of spotting the animals. And you can see here, these are the 3 matched pairs. So in orange is the ratio of juvenile to adult abundance in the reference population. So that's with the sham vaccine. And in blue are the ratios for the gonocon treatments. So you can see that there were a higher number of juveniles compared to adults in the reference stroke untreated population. And it looks like gonocon had an impact on reducing the cohort of new offspring. Here we just collapsed all the treatment and reference populations into just a binary method, and so you can see that the field scans of juveniles were much higher in the reference sites, and this was this statistically significant difference. So lower abundance of juveniles in the gonocon sites. So one of the rules of ecology is you should always stick with the results that show what you hoped for, but we also tried a different way of looking at the impacts of the gonocon. So we actually did recapture data. So we went out and retrapped animals in the summer following the application of gonocon. And we had some good news in that 2 of the sites in Hogbacks and Bluff Lake, you can see again that there are a lot more juveniles captured than in the gonocon treated sites. So in fact, no juveniles found in Hogbacks at all where we had treated, and only 1 in the Bluff Lake site. So it looks like there's a difference. Unfortunately, we had 1 pair where we failed to capture any animals at all. We still don't know the reason for this. It looked like there was some kind of road work nearby, but we have 1 matched pair where we essentially have no results. And so this has rather muddied the waters in one respect, but where we were able to capture animals reliably, it looks like gonocon did have the desired effect. So can we use gonocon as a potential management tool? A paper by Yoder et al. It's a really great look at how to combine these 2 approaches. And they suggested that an annual contraception of about 33% of animals should work with modeling and hypothesized impacts. Or you could try it less frequently, but at a higher rate. We feel we got a lot of at the site level that we were looking at, we managed to get a lot of the females captured. So this is feasible. And Yoder et al. Suggested that this might be used in combination with culling. So reduce the numbers, but also reduce the reproductive output of the surviving animals. I guess the difficulty is using this at the scale that works for land management. So discrete urban prairie dog colonies are often quite finite in their boundaries, and it is potentially able, we can capture all the females across the entire site, and that should be doable with some effort. The more rural or the increasing rural areas, it's unlikely to be realistic purely because of the difficulty in capturing animals. But it certainly would be feasible to do at the boundaries, and especially where you might have areas that you are worried about conservation-wise. The data we have acquired have been submitted to the EPA, and I believe gonocon is undergoing the early stages of product registration for use as a prairie dog population control tool. So that about wraps up the research that we've done. Again, I just want to thank the Botstiber Foundation and the Humane Society of the United States for their input. I believe these are links that you can look for their work in a more general sphere. And I think should be able to take questions. I guess I will turn off the screen.
You can leave it on.
Okay.
Since you're going to be answering questions. So. You may want to mute yourself while I'm looking through some of the questions we have. So I have one to start us off with. So what percentage of the population do you think that you were able to treat in your 2 treatment sites? Does that make sense? So did you get a baseline? Did you do a census to determine how many animals you were starting with? And then after that, were you able to determine within some range what percentage of the target population you were able to treat? Oh, turn on mute, unmute yourself.
Oh, still, after all these months, it's that classic.
So I'm going to mute myself so there's no feedback.
Okay, so yeah, we were often just using a subsection of the prairie dog colony. So it was how we were figuring about 100 traps so that we could get a reasonable amount of animals and yet not be overwhelmed. And where we did that recapture analysis, so getting the animals again, we found very few new adults. So we actually got a good proportion of the population where we trapped. But that was often a variable. So some of the prairie dog colonies were a lot larger. So you could see them increasing. So it was a small percentage of the overall colony site. And some of them were smaller. So we were getting a bigger proportion. So I guess the implication of your question is it feasible to get an entire colony? And that just depends on the actual circumstances. And that's why I was saying I think urban discrete colonies would be more feasible to work on than the more unimpeded colonies that are on the outskirts of towns. And that's what this work is always intended to be, was to look at urban management and not management in the rural.
That's a good point. Okay, so next question. Are predators affected by gonocon treated prey animals?
Sorry, gonocon treated?
Prey animals. So, so in this case, prairie dogs.
Yeah. So that's a great question. I should include that in the thing. So the theory is that because this is a reproductive hormone, if any ingestion of the animal, it should be destroyed during ingestion. So it shouldn't be an impact upon predators. And one of the things we did do was do this all manually. This was not a bait associated initiative, so that we could just look at the impacts of animals and reduce any side effects of the treatment. So I think the answer succinctly is there's not believed to be an impact upon predators.
Okay, next question. Were there behavioral assessments to determine if gonadotropin interference altered social structure?
Yeah, that's a fantastic question, and especially in this context when prairie dogs are such a social, behaviorally complex animal. And so we did a lot of head scratching about that, and we decided to just treat females because we didn't want to interfere or potentially interfere with the territorial behavior of the adult males. So I should just explain a little. The family groups of the prairie dogs, or blacktail prairie dogs, often an adult dominant male with some adult females and some younger females, and the adult male mates with those females, and then they have these kind of family groups called coteries, and that is what you have as a massive colony, is a lot of smaller coteries defending their territories within the colony. So we didn't treat males so that we wouldn't interfere with that. And I think it's a really interesting question on whether there are impacts on behavior. That was a little beyond the scope of this examination. We were just trying to investigate the impact upon the numbers and the reproductive output.
Next question. Do you know if the relocation works in the sense that animals never go back to the original place? So I guess in the sense of relocation, would they attempt to go, like some squirrels are known to, they have homing instincts that would, if you release them miles away, they may make their way back home. Is that the same in prairie dogs or no?
Yeah, I'm afraid I don't have a good answer for that. The numbers and descriptions of relocation efforts is harder to find than one might think. I think it's often in the gray literature rather than scientifically published. And there's often confusion as to what is the success of moving versus what is the result of the success of actually staying put over a longer term. And I think, I honestly don't know the answer. A lot of this is very complex habitat, so there are a lot of boundaries in terms of urban areas between location sites. But I have seen animals moving right through the middle of Fort Collins outside Ethiopian restaurants in strip malls. So these guys do have the potential to move, and they do so. It's just, I don't know what the impact is for relocation.
I think you answered this question. So does this drug impact the meat for those hunting prairie dogs?
Is that for people?
Yeah.
I mean, theoretically it shouldn't, because again, it's ingestion and or treat. If you cook the animal even better, I imagine that would cause any hormone to deteriorate. But I'm not honestly aware of many people hunting prairie dogs for food.
Okay.
That's my perspective.
And I also think that you already answered this question. Did you apply the treatment to both females and males? And you said females only, correct?
Yeah. So it was only adult females, and we also didn't do the juvenile females. And I think one of the, like a secondary investigation might incorporate the juvenile females also,
right?
Because they tend to reproduce that following year as they grow. But we looked at adult females only.
So this is an interesting question, and I was going to ask the same. I know the answer, but I know that there may be some progress being made on this. Is there a bait form, meaning an oral form of gonocon registered and available for use?
I don't actually know the answer to that question. I mean, for prairie dogs, that's not yet on the agenda, so it might be for other animals. Mm-hmm. And then I guess the corollary questions are, what then are the impacts upon other wildlife in those habitats? So given the prairie dogs are such a key integral part to those habitats, a lot of other animals living there that wouldn't otherwise, are there potential impacts upon other rodents that would eat those baits?
Right.
And
the next question is related. Does this treatment have to be administered via injection, or can it be used as an oral uptake like in a bait? And so you just answered. I will tell people that there was a paper that was published by one of our advisory board members and her colleagues, Giovanna Masai, where they're attempting to encapsulate gonocon so that it can be taken up orally and not be broken down so much. I think I'm saying this right, in the digestive system, where it would still be able to have the intended effect, which was, disrupting the reproductive cascade to cause infertility. So if anybody is interested in learning more about that paper, just shoot us an email and we'll be sure to get in touch with you about that.
And I should also mention, there is another fertility treatment that has been tested in prairie dogs that is associated with an oral bait. And for some reason, because I'm on a webinar, my mind is totally blanking on the name of it.
Is it
that's what I'm thinking. I just hesitated to be.
I'm not sure. I know what you're talking about.
Yeah.
So Again, folks, if you're interested, send us a question. One of us will track that information.
Right.
Okay.
So that has been used, but only on a single plot. So, and it seemed to be successful, but it does have those other implications.
And this is an interesting question too, a really good one. What are some of the local roadblocks, i.e. politics, that may limit the use of this product to manage urban prairie dog colonies?
I think one would be logistics. It is hard, you have to make that effort on the field to actually get all the prairie dogs that you're looking for. But this has actually been one of those happy kind of middle grounds. So because it's not lethal, advocates for prairie dog populations are happier than that extreme. And other folks, ranchers and cattlemen's association are apparently reasonably happy with this if it's used in the appropriate context. So it's not being used across broad swathes of ranching land, but it is, they have not had, they're not anti this kind of use also. So it seems in conversation, it seems to be quite an exciting prospect because most people are happy with its potential. And that the end situation is something we're all looking for, and this is being applied in a humane manner. So everyone seems happy so far.
That's great. Question on behavioral assessment. Would treating males with gonocon potentially encourage submission or higher chances of dispersion? That's question number one. And then would that be a favorable outcome in urban populations to get the prairie dogs to leave to rural places?
Sorry, was that the impact on males was the question?
Yeah, so would treating males with gonocon potentially encourage submission / higher chances of dispersion, and would that be a favorable outcome in urban populations in order to get the prairie dogs to leave to rural areas?
Yeah, I don't know the answer to that. One of the reasons we didn't treat the males was because we were worried about the argument that if one male is treated and becomes non-reproductive, you would only need one male in the area to take advantage, right? So that they would mate with just a larger number of females. And therefore, you wouldn't get the impact or necessarily see an impact from treatment of males that we see in the females, because if everyone is remaining in their family group, it's almost a surprise, right? As opposed to watching some males become dominant. I don't know about the impact on dispersion of males if they are less aggressive or less dominant. One of the issues would be, are you then exacerbating movement, and so you are creating new colonies in an area where people aren't necessarily wanting them? And that's another question that we would like to ask and answer is, what are the longer term impacts? So it looks like with some rudimentary scans, the gonocon doesn't seem to have a huge impact the following year. It looks like either new females are there and reproducing. So if you were to allow treated males, this is all hypothetical, to move somewhere else, then they would become reproductive the following year or the year after that, and then you are essentially seeding the population.
Yeah, I'm not an expert on this, but I think one of the potential upsides of this is that you're keeping animals in what is maybe considered a high tolerance zone, a place where people want to see them and be there to appreciate them. And the goal really would be to prevent dispersal into places that are low tolerance areas where people do not necessarily want them. And those may be some of those rural areas, if those rural areas are agricultural and not native prairie habitat, right?
Yeah.
So something to keep in mind for people thinking about this. Another question, was the registered product gonocon used in your studies or a different formulation?
This was made by standard operating procedure by USDA, so I imagine it's the same as other forms of gonocon. This was not modified in any way.
Okay. Next question. A person was wondering if the EPA registration you are pursuing, is it being registered for use just in black-tail prairie dogs? My understanding is the EPA label laws are very restrictive. That's a good question.
That is a really good question, and I can try and follow up and find out if that's the case.
Okay.
One of the issues being, though, that black-tail prairie dogs are the ones that are considered often problems, and some of the others are actually are wavering around conservation, different status for conservation. So Gunnison's prairie dog, for example, is something that I think it would be hard to start controlling because they're actually looking at protecting them.
Exactly.
So, and black-tail prairie dogs also are the most widely distributed of the species. So, but that's a really good question. I don't know whether the registration has been just for that species or for prairie dogs in general.
And then you may or may not know the answer to this question, but what are your next steps with your research, and what is the status of the EPA registration?
I believe EPA registration started in the middle of last year, or towards the end of last year, and I think it is normally around an 18-month procedure, but we shall see. And then I think some of the questions that you guys have raised just today, that what are the longer-term implications would be the next step, and whether this is workable or feasible or something we would like to use as an oral bait as opposed to something requiring vaccine administration by hand. So I think those are the next things.
And then to that point, so someone is asking, can Ronnie Rancher or Hannah Homeowner purchase and use gonocon, or is gonocon currently unavailable to the general public? It's a good question.
I don't know. I don't think they could use it without the EPA registration process having happened.
Right.
And then I'm not sure what the procedures are to administer.
I suspect that they would have to hire somebody that has a pesticide applicator's license first in their state, and then that state would have to have gonocon registered as a pesticide for this use. And then so on and so forth. So no, I think the answer to that question for now is a definitive no. But you know what? It does speak to, prairie dogs are a keystone species. A lot of people don't know that they don't exist on more than probably 90% of their historic range. And be that as it is, I think there are people concerned with doing anything to limit their populations when, as a general, thought, they're sort of an imperiled species. A lot of people don't think of it that way because if you live in Colorado, you think they're everywhere, but on an ecological scale, they're not, and they're not in the places where we need them to be in a lot of respects. So how do you explain to people that, number one, that the gonocon logistically, the way it's administered through injection, probably wouldn't be able to treat a lot of prairie dogs to begin with, that it is really meant for these little discrete colonies. But then, how do you address concerns about using anything that would limit population growth on, like I said, an animal that a lot of conservationists believe is an imperiled species?
Yeah, and I think that's exactly the issue. So one of the reasons, or one of the rationales for this was that urban situation where land managers who are conservation oriented, who want to look at intact ecosystems, but were like putting their hair out because the problems associated with their dense urban prairie dogs, their dense urban black-tail prairie dog populations were being exacerbated and causing more problems than existence. So they were having issues with local homeowners and having issues with erosion of the property. And for example, here in Fort Collins, we have issues with them encroaching on endangered species of plant that they're overgrazing. And so all we are trying to look at is that place where the problems are becoming dominant, and that you're in danger of having control at a greater extent. So I know one of the land management boards were really upset when they had to resort to lethal control just to get the population under control. And it's a constant struggle for them. And so what we were hoping is that this could be used as a way of getting that win-win. I don't think the intent for this is to use it on, not my intent, and I know these things have potential ramifications beyond the control how and where you use it once registered. But I don't think this is going to be one of those mechanisms where you would control it in the larger rural system. In part because at the moment its application would just be unfeasible. If you're looking at colonies that are 500 hectares, it would take so much effort to actually control them. Whereas we were looking at colonies that you can walk around in 30, 40 minutes.
Exactly. So a couple more questions. Thank everybody for the questions. These are excellent. Perhaps it has been mentioned before, but I may have missed it. This is from one of our participants. In the study on Norway rats and gonocon, how did they administer gonocon, and at what frequency do you have to administer gonocon for it to remain effective?
I don't remember the details from the black rat population. We made a single administration of the vaccine. So this was done before, or as much as we understand, before the breeding cycle begins. And so we were doing this before the hormonal cascade, so late summer, early fall. And we made a single vaccine injection for all animals, and that was effective, or at least seemed effective in reducing the output, reproductive output the following spring. And like I said earlier, one of the questions is how long this lasts, and whether this the same females are still not reproductive in a second year or not, and we're not quite sure of that data, and that'll be one of the things to follow up on.
Okay, so this may or may not be a question that you can answer, but give it a shot. How does the cost of the gonocon program compare to lethal and other non-lethal solutions?
Again, so that's more the economics, and I'm not too familiar with the poisoning, how much it costs to poison. I mean, I would, in an uninformed way, I would say probably poisoning is cheaper, but I'm not sure that the long-term implications are better. That you get this rapid recovery of the thing, so you have to keep on poisoning, and that has all the downsides associated with human health. So I think gonocon, at least when it's not commercial, is not a very expensive vaccine, and so feasibly that can be done in a local small population, especially if you can get local involvement by groups to actually administer it.
Like volunteer.
Yeah, and we had lots of people who wanted to volunteer. Field assistants were not hard to come by. It's just a case of on a proper management scheme whether that would work. But I think in this kind of middle area of finding long-term sustainable management and cost, that this seems to offer a good opportunity.
Yeah, it sounds like the situations that this is intended for, that tends to be what happens, is a local group that is interested in doing something different. They often are very interested in being really part of the solution if they're given the opportunity to help in some way. Yeah,
yeah, and just as an aside, like it's not a field method that is overly hard, right? It's catching prairie dogs doesn't require like possibly losing limbs. They're manageable, and you can do it with volunteers. It's something that's achievable.
All right. If gonocon must have a repeat use, do the prairie dogs get trap shy, making it more difficult for continued use? That's a good question.
Yeah, I think prairie dogs, just like anyone else, have different tolerances of the traps. So some of them are trap shy, I think. Some of them are trap happy. So we would catch the same growingly obese females day after day who are just thinking, great, we've got this horse bait ferret day. So some we definitely recaptured with almost annoying frequency, and some we didn't see again. But it looked like by looking at the traps in the same area, you did get a good handle on the actual local prairie dogs.
Imagine if the experience wasn't too terrible, that you can understand why they would keep returning, because it's good food that you're putting out there compared to what's available to them that they're competing with others for. So
and using those handling cones, like it's amazingly speedy. So especially if we had caught one before, they would be out of the trap in less than a minute. And so not having to use anesthesia for this process definitely helps with the logistics of being in the field and recovery of the animal. So you don't have that that worrying thing of whether you've given too much anesthesia and whether they're going to recover appropriately. They are conscious throughout the process.
And then the risk to the individual because of not knowing how they're going to process the chemical immobilization drugs.
Yeah.
Is gonocon registered in Europe?
I don't know.
I don't either.
Prairie dogs aren't there though.
That is true. There are other rodents there though. Who is registering it for prairie dogs? Is it the NWRC, the National Wildlife Research Center? Yes. Okay.
Yes. Yep.
Somebody asked us that is this recording going to be made available? The answer to that question is yes, we're recording it and we will be posting it on the BIWFC website soon. Do you think that this could be used as a conservation tool to reduce densities of colonies and to help reduce the likelihood of a plague outbreak?
That is a really fascinating question. So, and that's partly why I got interested, because my background is more in the ecology of diseases like plague and prairie dogs. So yes, I think the conservation tool for abundance and keeping densities lower and not exacerbating problems is the key aim of this. And in terms of the academic interest of whether this can have an impact on plague dynamics is really interesting and something that could be used. Again, the corollaries for that is often if you're looking at plague management, that is in part because you're interested in black-footed ferret persistence, and they might require a higher density of population of prairie dogs. So a lot of trade-offs and unknowns, but a really interesting area to think about.
Yeah. I hope I'm interpreting this next question correctly, Dan. The person is asking, what stressors will a lower prairie dog population have on the ferret population?
Yeah, so I think that we just briefly mentioned that. I don't know what the black-footed ferret requirement is for density or abundance. So whether a larger but less dense colony would do just as well as a smaller high-density colony. So I'm not sure what the impacts are.
Given the intent, like I said, it's really important to emphasize the intent of this particular technology in these urban systems. I don't know enough about black-footed ferret populations to know, but since they are a highly endangered species, I don't know if the U.S. Fish and Wildlife Service or Colorado Parks and Wildlife know where many of them are, and therefore, if they even thought that this was an area where they may be present, may not even want something like this used. Neither would somebody that was proposing to use it if they thought that density might have any impact on the possibility of sustaining an emerging population of black-footed ferrets. So,
yeah. And also the reintroduction program for those are in much more rural systems. So this is not supposed to be something that overlaps. Because black-footed ferrets don't do well near humans, don't do well near dogs and that kind of thing. So that it's if you were to choose a spectrum, they're at the very far end of the spectrum of prairie dog urban situations and control. So you're looking ideally for the most sustainable and robust prairie dog populations you can, and so it's not an instant overlap. I think the questions are great though in just terms of thinking about the potential ramifications.
How were captured birds marked, if at all?
So we just marked them with a pit tag. So it's just a little chip that you inject in the scruff of their neck, like you do for dogs or cats for identification by your vet. So that's in terms of capture, that is really fast because once the prairie dog enters that handling cone, you can scan it with the scanner and it will pick up the ID. And then the prairie dog can be released, like I said, within a minute of arriving at the trap and released. So it's really fast. They can be marked with ear tags if you're looking at that kind of thing, but there's often an issue of them tearing them out. And so you're reduced to just going, oh, this is an animal we obviously know we captured before, just because of the tear in an ear. And then one other method that people do use is using dyes, but that has an implication that once you have the molting season, which occurs between administration of the vaccine and when you would look at the impacts, you would lose the identification. So that's why we chose the pit tags for a longer lived ID system.
And somebody in Europe said that in Spain, gonocon is not registered, but they want to register it for wild boar.
Oh yeah.
And then they said, somebody else said it is not registered in Europe as far as they know, at least not for wildlife in the Netherlands. So those are the questions we have so far. If anybody that's participating would like to ask Dan a question, please feel free to go ahead and submit one in the Q&A. We'll sort of keep this going for a few minutes longer if anybody has any other ones. But while we wait to see if anybody is going to submit one, Dan, thank you so much for your time. And thank everyone else for the questions. That's really what makes these webinars really, really awesome, is getting people like Dan to give feedback on their work. Oh, we do have some more coming in, so we all really appreciate that. So a comment for Dan: I work with the Utah prairie dog threatened species. We have large colonies on federal lands that every few years succumb to a plague outbreak once densities have gotten high. We are starting to trap and relocate animals out of the colonies. So just letting you know that. Somebody else commented that EU requires GMP manufacturing. It's currently not manufactured with those requirements, so that may be one of the hurdles to getting it registered in Europe. And then somebody added, and it might be interesting that this could be used as a tool for a threatened species and not an animal that's more ubiquitous, like black-tailed prairie dogs. And then somebody wants to know, what type of bait did you use in your traps?
It's horse sweet feed, so it's just a pet supply, feed supply kind of horse food. Yeah, but prairie dogs, you can use a great deal of things. You could use peanut butter or oats or whatever. The key seems to just be having traps out for a little while so they get used to it.
So they have to be, there's an acclimation period for you start.
Yes, yeah. So we always put traps out 2 weeks before we actually plan to start trapping. And then they're wired open and we put the bait in every 3 or 4 days just to get them used to it.
Very cool. Were there any sites where your bait was not working and you had to try something else?
We just had that matched pair where it worked the first time in the fall, and then we just could not capture prairie dogs the following summer. We don't know why that is. We don't know why it is. So everywhere else seemed to work.
Interesting. Okay, we've got about like 7 minutes left. If there's anybody else that has any questions, please feel free to submit them. But if not, again, Dan, thank you so much for your time. Monique, do you want to chime in, or?
Yeah, definitely. If there are no more questions, I'm giving it one last check. No, just nice comments. That's good too. Well, thanks to Dan.
And thank you guys. This has been a really tremendously interesting project, and it's good.
I know Dan probably cannot see some of the people that are commenting, and I know some of them, but I just wanted to give a shout out to Dr. John Hooglin. He has done some of the most groundbreaking work on prairie dogs, prairie dog ecology, populations, etc. in the world. And he was submitting some really thoughtful questions, and he just wanted to tell you it was a wonderful presentation, very informative, lucid and provocative, and to keep up the great work. So I thought I would pass that along to you. And it is our honor and privilege to have had Dr. Hooglin checking out the webinar today.
Yeah, thank you. That means a lot to me. Thank you.
Yeah, I'd just like to thank you again, Dan. Thank everyone that attended. Thanks for all the great questions. They really were very good questions and comments. And that our webinar series continues. Our goal is to hold at least one a month. So check our website, wildlifefertilitycontrol.org, for continuing webinars like this. Our next webinar is actually next Thursday, February 4th. It is a webinar that Stephanie and I will present about our new grant guidelines. So if you know anyone that would be interested in that, be can check that out on the website as well. And I think that's everything.
Too.
It was a great webinar, Dan.
Thanks, guys. Thank you very much.
Seeing
Thank
you.
you to everybody for attending today. We really appreciate it.
Yeah, thank you.